Okay, good afternoon, guys. Again, my name is Melvin Simoyi. So today we're going to cover the genetics part lecture, on chapter 25. We're pretty much going to, let me see if I can use, there you go. Okay. So, genetics and human disease, right? This was started a long time ago, as you all know, by a geneticist called Gregor Mendel, who used to live in what is now the Czech Republic. In the later half of the 1800s, he discovered that, of course, biological traits are, inherited by the offspring via independent units that we now call genes. All right, next, we're also going to cover some genetic diseases today. All right, these genetic diseases can be inherited. These genetic traits can be inherited. But I wanted to point out that as we talk later on about genetic diseases, genes do not cause disease per se. Okay, only when there's a mutation does a gene cause a problem. All right. So, and there are various mutagens, that could produce these genetic mutations. We all know one of them is viruses, right? Okay. What else can cause genetic mutations? Yeah, radiation. What's that? Yeah. Yeah, that's a mutation. Yeah. Mm-hmm. And yeah, somebody said chemicals. Okay. And all these, can cause, genetic diseases, and they can be inherited. And later on, we'll talk about chromosomal conditions. Those ones are not inherited like the genetic, the mutations, because they are actually problems with the whole chromosome. And that happens during mitosis when meiosis, when the gametes are being formed. So we'll talk about trisomy and monosomy and so forth. All right? So good. Next. I don't want to punch the wrong button here. Okay, so we've got a question now. I'll probably just ask a question and then talk about the material. Ask a question and then talk about the material. All right? So we're going to, use your little clicker thingies. So, and then when I start the polling, it will give you like 30 seconds, and then we can just talk a little bit about the question. All right. So start polling. Is there 30 of us or 32? 31? 32? Somebody who's not here, maybe? I'll just let it time out. Okay. And show results. Okay. So now we go to the answer. Okay, there you go. Okay, so 68, more than half of you got it right. It's a protein, because we have transcription in the nucleus, right? And then that's when the genetic code is copied onto a messenger RNA molecule, which exits the nucleus, gets into the cytoplasm, where it will be translated into a protein, all right? And the protein, in fact, when we next we'll be starting to talk about gene mechanisms and how they affect the cell and the functions, all right? And it's all to do with proteins. All right. What's the main substance that proteins represent in the body? I'm not talking in terms of muscle, but in terms of a functional compound. In terms of a functional compound, what do proteins make up? Enzymes. Yeah. So enzymes are the way the genes actually affect the functionality of a cell. And also structurally too, because enzymes I mean, proteins are also components of the membrane, components of the organelles, and so forth, right? Okay, so I hit hide results, and then I'm supposed to click in here. Okay, to go back, and then next. Okay. All right, that worked nicely, so I'm happy about that. I usually have problems with, technology. All right, we just talked about how, of course, genes, genetic information is inherited in genetic units called genes, and they carry the genetic code. Actually, the codon, how many bases are in a codon? 3. Like, say, ATT, added in time. This is just an example because I have AT&T for my provider. Okay, but so they go in triplets. All right. So what happens, if you have a single gene mutation where you have one base pair is changed, it affects all the triplets, and then everything is going to be out of whack. That's why just missing one base pair will disturb the whole protein. Because how does a protein fold up? How does a protein fold up into this tertiary structure? Well, yeah, it's those interactions, hydrogen bonds, but it's the R groups. The R groups on an amino acid determine how the protein will fold up on each other. So if the amino acids are in the wrong sequence, it's not going to fold up into the proper tertiary structure for that particular protein. All right. So that's why genetic mutations can be really bad. All right. Because the way genes affect the functionality of a cell is that we talked about it earlier. Proteins, make up the enzymes. And what do enzymes do? Enzymes, they determine the speed of the reaction. They also determine what kind of reaction occurs. All right. So that's the functionality of it is affected by that. Besides just catalyzing and determining the kinds of reaction happening, what else? We said there are structural components of the cell. Where can you find proteins in the cell? Which parts? Cell membrane, yeah, which is very good. And we'll talk about one genetic condition that occurs when there's a problem with some sodium ion, chloride ion channels. All right, in a little bit. And then the effects of that will be profound. And then also structurally as parts of the cells, organelles, and we're going to talk about, meiosis, and we going to need to know about the tubulins, which is a protein that makes the cytoskeleton of the cell, and it actually, and also the formation of the spindle, all right, and that is going to affect how the cells divide up, and it's going to affect chromosomal conditions, and we'll talk about that as well. All right. So, all right. So DNA actually does not occur naked like that, right? What is chromatin? Chromatin. What is it? What is floating there?
Oh, just the strands of genetic, DNA.
DNA, yeah. Mm-hmm. But what is it made up of? What is chromatin made up of? DNA, you just said it, and what else? Proteins. Chromatin is actually DNA and plus proteins. Okay? And then the chromosome is actually folded up part of that, of the genetic material. And it's inactive because it's really folded up. All right? But chromatin is in strand form. Okay? And that's what becomes active, and that's what is transcribed. Okay? Why would you think that DNA is attached to these histones, these proteins, instead of just appearing as DNA? Wouldn't it make it easier for transcription and so forth? Why is it all coiled up and wrapped up in these proteins? What do you think is a good reason for that? I'm talking more in terms of functionality kind of thing.
Isn't it for regulation of proteins are expressed and then they
Yeah, that's part of it. Some of the histones actually determine how, but I'm talking in terms of something that causes problems. But it's true. The proteins, they control what genes are transcribed, and they regulate, which gene is going to be transcribed. So that's one. But, if DNA is just out there for all these elements that we just talked about, viruses, radiation and so forth, to attack, you have much more mutations than you can handle. So the fact that you have the protein wrapping it around, even the active form is not just DNA by itself, right? These proteins are necessary to protect the DNA from mutation, all right? Okay, and even when you're transcribing the DNA, what happens is that only that section is unwound, because DNA is a double strand, right? So only that section that's actually being transcribed is unwound, because it's being exposed when that happens. Okay, so I just want you to note the importance of the proteins. All right. All right. So. Okay. So next. Oh, question. All right. So I will start polling. 30 seconds. Oh, there you go, right on the last dot. Okay, I like the good thing about this is that it's anonymous, I like that too. Yeah, I bet you do. Okay, wow, we got a variety of answers like a rainbow. The sun is not out, but Okay, good, and I like that. That's good. That's why I asked the question before we go through the information. So. Let's see what the answer is over here. There you go. There you go. There you go. So it is about, there's about 20 to 25,000 genes in the human genome. Okay. And the Human Genome Project actually just released this information, I think 2003. So they've been haggling over it. Trying to find out how many genes there are, just to the whole genetic material in the human. And this is actually a quarter of what they thought. Actually, it's a quarter of what they thought, this sort of 20 to 25,000. They thought it was 4 times that. And also, the fruit fly, we have only 1.5 times more genes than a fruit fly. All right, so yeah, that's something. A fruit fly is like, and it's so and you can imagine us and a much less percent, which we will talk about in a little bit, is actually coding for proteins that we've just been talking about. The enzymes and so forth. All right. So okay, very good. So we go. So how many did we get? Oh, more than half. At least more than half are getting it correct. So that's good. All right, so what do we do next? Hide results. And I'm supposed to. There you go. All right. Okay, so the human genome. Of course, the human has got 46 chromosomes. So it's 23 pairs of the chromosomes. All right, and they're attached by the centrosome in the middle there. Okay, but there's also one chromosome that occurs in the mitochondrial DNA. All right, and this mitochondrial DNA actually creates some diseases that affect males mostly, because and they get it from the mom. Why? Because when the gametes are being formed, what happens? You have the egg cell, which has got all the nutrients in it and all the organelles in it. And then you have the sperm cell that only the head enters. And just a bit of the mitochondria. Because it's got a meat piece, the sperm cell, it's got a meat piece which is made up of mitochondria for swimming up the reproductive tract of the female. So most of the mitochondria that are contributed to the zygote formation come from the mom. So most mitochondrial diseases, inherited from the mother through the mitochondrial DNA. All right. There's some neuropathy, muscular kind of dystrophy conditions, like one called Miller's. It's mitochondrial encephalopathy, lactic acidosis symptoms. So you get tired and so forth, and it's comes from that, and it comes from the mother. So the sequence was roughly in the 2000s was elucidated with the Human Genome Project, and it's about 20 to 25,000 genes, right? Okay, does anybody know what percent actually codes for proteins? Yeah, less than 2%. Less than 2% actually codes for protein. Let's just say 1%, which means roughly about 250 to 300 proteins. Okay, and we'll soon be talking about the proteome, which is all the proteins that is coded by the genetic. All right, so there's only 250 to 300 proteins that is actually coded for. All right, so we got all this 98% of genetic material. All right, all this 98% of genetic material. It's sometimes called junk DNA or non-coding DNA, right? Okay, before I talk about the pseudogenes, does anybody know, or can somebody say what a reason is to have all this much genetic material when we only need 250 to 300 genes? Yeah.
To turn on and off certain genes or expressions.
Okay, very good. But 80% of the non-coding DNA turns genes on, off. All right. What about what other things? Yep.
Could it afford protection? I mean, if you have a certain number of sequences that are before the actual protein, it increases the chance that maybe one of those gets hit, and it's a kind of a who cares, it's not coding for anything.
Protection against what? It's okay. I mean, it's good to think like that because that's how everything. Yeah.
Basically, the extra sequences that are on there that don't.
Yeah. I can see what you're saying. It protects the genetic material that actually codes. The likelihood, yeah, that's actually a good reason. I didn't think about that. If you have many, the chances of you hitting a gene that actually codes for something is less. It's just like winning the lottery. The more people you have in the pool, the chance of you winning is less. Yeah. So same thing. The chance of you hitting a gene, because there's just a lot of it that actually works for something, is less, so you have less mutations. Very good. What else?
A lot of our DNA, I believe, is viral, leftover viral DNA.
Well, yeah, you might be onto something. Yeah, because viruses can inject which is what the basis of some of the treatments we're going to talk about is. Yeah, that could be a thing. But what about adaptation? Okay, I'm sorry, my handwriting is really neat. Adaptation. But when you see this, you'll be able to see and hear what I'm saying. So you know it won't be adaptation. When an organism is going through the motions and adapting, there will be certain genes that may need to be switched on. Okay, so if they're not there, then tough luck, and it's survival of the fittest, and you want. So part of having a lot more genetic material than you actually need is so that when the environment changes, then you could switch on certain genes that were not necessary at that time. All right? Okay, and another thing about this is pseudogenes. As we evolved, we started as Homo habilis and Homo erectus and so forth, and we were going, and we had certain genome then. So the pseudogenes will be remnants of the genes from our ancestors as we were evolving. Okay, so it's a small percentage of that called pseudogenes. And these are actually broken and they don't actually work. But the 80% is, of course, switches on and off genes, which is very important. And then adaptation, you can adapt, because you can switch on certain genes that you didn't know you needed because the conditions change. Like right now, we're going through climate change. Who knows, we may end up being aquatic animals again once all that ice melts in the poles. We need to swim again. You see? So all that, because we did start as fish, and then we evolved into tetrapods that lived on the earth and so forth. So we might just go back. That's where I think it's heading with all this global warming and all this melting of the ice. Once that ice melts, it will really flood the whole earth. Besides, we are 70% water anyway, right now. Okay? All right. So, okay. So this is a karyotype. A karyotype is literally what? A picture of the chromosomes in your nucleus. That's what it is. They use an electron micrograph, which is very high resolution, and high magnification. So what a karyotype is that they take pictures, electron micrograph photos of the nucleus chromosomes, and then they order them from largest to smallest. So chromosome number 1 is the largest chromosome, all right? And then all the way to the smallest, which will be the sex chromosomes. And we'll talk about those in a little bit, all right? So that's what a karyotype is. Can you sort of tell me what kind of conditions we could diagnose using a karyotype?
Trisomy 21.
I like that. Yeah. And what kind of condition is that, trisomy 21?
Down syndrome.
Down syndrome. It's what group of diseases does that fall under? Chromosomal diseases, yes. Okay. You cannot diagnose genetic single gene diseases with that, right? Because you'd have to see the gene sequence, And so the karyotype doesn't do it for that, all right? So we have other ways we could determine that, all right? But anyway, and then an ideogram is just a cartoon of this, chromosome showing this centromere, all right? And what about the bands? There's something I want to talk about when we start to talk about P and Q arms that I'll mention later on then. But these bands, what do they stand for? Bands. These bands, this rainbow-like bands on the chromosome. What's that? No. Yeah. They signify the location of genes on that chromosome.
So do they have special dyes that dye them all separate colors like that? Or?
I don't know how they do it. This is like a diagrammatic form. I'm not sure what kind of color it comes up on the actual, because I think the it's black and white, electromicrograph. So this is done for you to, as a diagram purposes, really. Like sometimes they show falsely colored sperm just so you can see, but it's really colorless. But yeah, these bands, they can actually see this dark and light on an electron micrograph picture, I think. But they are very important in determining the exact location of the gene. Okay, and the geneticists, they use the centromere and use the P arm and the Q arm. So the P arm is the shorter arm of the chromosome, and the Q is the longer arm of the chromosome. And they judge the distance of that gene relative to the centromere, which is right there. Okay? All right. So the karyotype, we'll talk a little bit more about it later on, but it's used to diagnose these genetic diseases like the chromosomal disease. All right, and then we're going to go through towards the end pictures of all these. We'll show like the phenotype of that disease and then the karyotype, and then before I put the name of it, you guys, I want you to determine what genetic condition it is. But we will have talked a little bit about it, okay, using the karyotype. All right, I can use this. I'm used to going to the board. Oh, there you go. So genomics, of course, the study of all this science of inheritance and the genome, all right? Okay, and then proteomics, of course, like I was saying, it pretty much studies all the 250, 300 some proteins that the genome codes for, all right? We understand just less than 2% actually of the genetic material codes for proteins. So the study of those proteins, all that group of proteins, it's very important to know these proteins, their functions, because those are the proteins that determine disease. All right? It's a good thing there's not a lot to study. You know, like 200, it's a lot, the 250, but you know, you could actually determine the sequence and the tertiary structure of that, and that helps you with, you know, determining you know, diseases and so forth. All right? All right. So, another question. Oh, I've got to start polling. Psh, I thought it was automatic. Yeah. It's got auto power off. Oh, 25. Oh, stop. You know, if you run out of time, you know, just press any answer. It doesn't matter. You know, we want to know who you are, you know, so it's okay. You know, just guess. That's what, you know, it bugs me when I'm grading exams and I see a multiple choice question and somebody doesn't check something. I mean, I'm like, there's one chance in 5, you know, you get it right. If you leave it blank, come on now, you don't do that. You never do that. Oh, okay. Actually, it's the, show results. Oh my God. Yeah, show results. Okay. People are liking P, which is good. Yeah, it's the P answer. There you go. Oh, there it is. Yeah, so it's the P arm. That's the shorter segment of the, and the longer segment is the Q arm. P, you know, stands for petite, you know, friends. So, and then they said the next in the alphabet is Q, so Q. Yeah, that's really what they did. But you notice that chromosomes are always like this. Right? Something really bothers me on this. You know, the location of the centromere. Does it bother anybody here? I guess I'm the only weirdo. Yeah. You know, why is it not central? You know, why is it not central like a bow tie? You know, like I'm saying. You know what I'm saying? Why? So I was like, this is bugging me. So I went online. I really looked and looked and looked, and nothing came up until I came to something that I was talking about, a chromosomal disease called 18p. It's actually tetrasomy. I know today we'll just talk about trisomy and monosomy, okay? Tetrasomy. And you can judge by the name, how many would we have? 2 pairs, right? Yeah. So it turns out that people with tetrasomy have a whole host of diseases. They have the regular chromosome 18 with a P and a Q arm, a short and a long arm, and then they have what's called an isochromosome. Just by the name of it, you can begin to see what kind of chromosome that is, right? It's got 2 P arms on it. And it's central, like the bow tie that I wanted, right? It turns out that's not good. It causes a whole bunch of diseases. So that's why this, I don't know why, again, it doesn't come close to a reason why the centromere is not central, especially when it's called centromere, right? You see, so that's why it bothered me. But that's the reason. It's because if it's on the center and you've got an equal either 2 P arms or 2 Q arms, you get problems. It gives a whole bunch of diseases. And I was looking at the diseases. Some of them I can't even pronounce the names right now. I'll be biting my tongue. They cover almost all the organ systems of the body. All right? So it is important because, when I'm teaching anatomy and physiology, I'm always telling them, my students, that nothing is ever the way it is in the body for no good reason. There's always a reason for it. So whenever I see something, I try to find out why that is, So that turns out it's actually a big thing that you have a short arm and a long arm. Okay, so if you get a chance to look it up, the chromosome 18P, all right, it will come up. So that's the reason why. So anyway, but it was really bugging me. Don't show results. And then I'm supposed to click in here. All right, very good. Too many buttons, then I gotta make sure I press the right one. So expression of genomic information, it can be expressed in various ways, all right? We just talked a little bit ago about the ideogram, which is a cartoon of the gene, of the chromosome, and then because the bands on it show the position of the genes, which is very important. If you're targeting a specific gene on a chromosome, you want to know where it's located so you can fashion a treatment on it, right? So that's very important. So there goes that short and long arm, the P arm, which is the short, and the Q arm, which is the long arm. And it turns out that the location of the centromere is not by accident. If you have equal arms on a chromosome, it causes a whole bunch of conditions and diseases in the human. All right? So it needs to be one long arm, one short arm. Okay? All right. And of course, the other representation is the karyotype, which is a picture of the actual chromosomes. The way you can actually see the, diagnose genetic condition, chromosomal conditions. All right? And also they can be expressed as the sequence of the bases on the nucleotide that make up the nucleic acid. All right? So the A is for adenine, and then thymine, guanine, and cytosine. All right? So adenine and guanine, oh, crap. I like this, the smart board, you can erase stuff. I really like that. Adenine and guanine are called purines, yeah. And they got this kind of deal, which is like uric acid. And that compound I really studied a lot in my graduate studies. So purines are longer. They got it's a longer base. So the purines will pair up with the shorter ones, the cytosine and thymine are the pyrimidines. Pyrimidines. So you will never get like a purine base pairing with another purine because they will be too long. You see, because it's a double strand, it's a helix like that. So if you have two, purines, it's just too long. If you have just a pyrimidine and a pyrimidine, it won't touch. They won't. You see, so it has to be one. For each nucleic acid, you need a purine pairing up with a pyrimidine, right? So, given these bases, what nucleic acid is that? Huh? Yep, it's DNA. And why is that? Exactly. Wow, that's good. And that's I'll see your name later. But yeah, the pyrimidine in RNA is uracil. Okay. So if you're seeing something that has a U on it, you know it's not going to be DNA. You know it's going to be RNA, right? If you see something that is all this AT, TT, if you see the T, like the AT and T I drew at the beginning, you know it's DNA. Okay, so that's how you can tell the two. Ribonucleic acid and, deoxyribonucleic acid. All right, so let's go. Oh, question. Oh, I got to start polling. I still haven't gotten the hang of this. Oh, crapper. No, that's not how I'm supposed to. That's the old one. Start polling. You see, I told you I didn't get the hang of it. I hope it's not going to mess up my thing now. Okay, let me see here. Oh my God, I pressed the wrong button. Only once, I don't get any forgiveness. Oh, wait a second. Somebody got beaten by the machine. Now I can show results, right? Show results. Everybody should get this here for sure. You know, that process is, of course, meiosis. Because meiosis occurs in two sets of divisions, right? The first one is a reductive division, and then the second one is really a proliferative division, really. It's like mitosis, just to increase the number of gametes. It only works for the male gamete, though, but the female gamete, only one egg comes out, and the four will be just the polar bodies, right? The reason because the egg, the ova, it's got all the nutrients, and then the mitochondria that I was talking about. So all the organelles and all the goodies go to that one ova, because it's going to supply the nutrients for life in the first week or so before the zygote or the blastocyst implants onto the uterine wall, right? Okay, so it's of course meiosis. All right, so we'll talk about a little bit about meiosis. Hide results. All right. Next. All right, so distribution of chromosomes. So we just talked about how you have the first reductive division in meiosis 1, which is meiosis 1. It gives rise to the 23 chromosomes, all right? These then become diploid again at conception when the male and the female gametes unite to form a zygote, which will have all the 46 chromosomes, all right? So we just looked at the karyotype just now with all the pictures of the chromosomes, right? And they were in pairs. So there's 23 times 2, which is 46 chromosomes, all right? So for the most part, there is what are called autosomes, right? So what are the autosomes?
Anything but the sex cells?
Yeah, anything. And what's special about the autosomes? The chromosomes are equal. The pair of chromosomes, they're equal, right? Autosomes, it's got equal in length, you know, chromosomes. This is my chromosome shortcut, X-somes, chromosomes. So, and then the other ones, like you said, sex chromosomes. All right, and these sometimes may not match. The other one may be shorter. In which sex is one short? That would be chromosome 23. What's that? Men. In men. Why? Because men have X, Y. And females, so this is a shortcut for male, and females have XX, right? That's why all the sex-linked recessive traits usually hit the men more, because they don't have the other pair of the chromosome to oppose it. You see what I'm saying? What's happening on the other X chromosome. Okay, so literally there's one like that, okay, and then the other one is short, which will be Y. So what genetic representation can you use to determine sex? That we've just talked about so far. We talked about ideograms, we talked about karyotype. Because you will definitely see the X and the Y or the two X's, which will be the same. So you can also diagnose, well, not diagnose, it's not a condition to be a male or female. Although females like to think it's a condition to be male, you know what I'm saying? Don't you think? But one of the things that I didn't actually, it is a condition. But actually, you know, men and women are really wired differently, for sure. We are totally different creatures. We are the same species, but we think differently. Actually, I was online the other time, and there's actually a video that went viral about the way, it was a comedian, about the way male and females think. Okay? With females, they multitaskers for one, and don't worry, I'm getting to something with this. Yeah, they're multitaskers, and everything is connected to each other. It's like their brain is like a mishmash. I mean, not a mishmash. That's not a good word to use. It's just, everything is connected. But whereas with men are compartmentalized. Yeah, you know, they've got different boxes. That's what the comedian said. And men will pick from a certain box and talk about only what's in that box. Nothing else. But female, everything is related. If you are late to come to my appointment here, it means you don't love me, and it means, you know, that's how the brain of a woman thinks. It doesn't mean, they won't think that, oh, it was traffic. That's why you were late. You know what I'm saying? They'll think, oh, it's because you don't care. That's why you know what I'm saying? Don't you think that's got to do something with the XY?
Absolutely.
It does. You know, if we cannot multitask, you blame it on the Y chromosome. Why? Because the man, it's shorter. So that means they really have less genes to pull from. So that's why we can only do one thing at a time. You know, focus. But women is like they got 10, you know, like an octopus with 8 arms. They can be doing this, you know, questioning the kids on this side, and, you know, but men can't do that. So, and I think the reason why we're different is because of the X and Y chromosome. Right? Don't you think so? It makes sense. Yeah, it does. And it probably is, because the females have more, you know, chromosomes, you know, the X and Y. So if there's a recessive trait here, there might be a dominant trait here, and then they're conditioned. So that's why most of these bad conditions mostly appear in men, because of that. Because there's nothing on the Y chromosome to oppose what's up here. All right? Okay. Next. Oh, this is the meiosis. And then, of course, you have the first reductive division, and then the proliferative division, right? All right. And this is only works for the male gametes, but the female gamete, only one comes out. But what this is showing is the concept of independent assortment, right? So these chromosomes can associate with any other chromosome, so that the thing that you want to achieve here is that you have different gametes, you know, so that each gamete is unique. It's got its own unique genome in it, okay? So that increases the genetic variation, and we'll talk a little bit about genetic variation and how it occurs and why it's important in a little bit. But the meiosis, of course, ensures that you have different kinds of gametes here, and you have other, you know, different kinds. All these are different kinds, and so that each individual is unique. All right. They have a unique genome. All right. Oh no, I pressed the wrong button. Okay, there you go. All right, so that's the concept of I was talking about a little bit. You know, genetic variation among offspring, which is very important. And one of the processes is independent assortment of chromosomes during gamete formation that I just showed in that diagram. Also, the genes can associate with other genes in the process of crossing over. You know, when the two homologous chromosomes are next to each other, you know, they can swap parts of the P or the Q arm, and then you have a different chromosome altogether. All right? What would you think is one of the reasons why you need to have all this variation, genetic variation? What one good reason for it?
They don't all look alike.
Huh? Well, it's you want to look like your mom or your dad. We may look like each other, but we will have our own genetic information in it.
Survival
Yeah.
adaptation.
Yeah, well, yeah, that's adaptation too. Yes?
Isn't it to create genes that, if a recessive, from like a PKU were to come by,
Yeah.
something would develop into which you can mask it?
Yeah, you're on the track, on the right track. What happens is that, like the lottery thing I was talking about, if right now they were to say, because Zilla, I live in Zilla, and it's like, what, 2,300, 3,000 people. If they said the lottery was only going to be held in Zilla, the chances of me winning would be very high. I'll definitely buy like a 50 or so tickets. But because the lottery is nationwide, people in Kentucky or wherever can buy these tickets. And so the chances of winning are much less. So if you have a lot of genes, a lot of variation of the genes in the pool, then the likelihood of having a recessive trait like it is much less. It's just numbers. It's a question of numbers. But if you have a limited gene pool and you have less, then the occurrence of recessive traits will be much higher. That's why they don't allow incest, because that's one very good way of limiting the genetic pool. Like if you have sex with your daughter or whatever the case may be, or your cousin, there's no other genes coming from outside. And therefore, that's when you have more recessive traits like Down syndrome and things like that get actually, okay, because then you have limited the number of genes, so the frequency of which recessive traits show up becomes higher. So genetic variation is very important for that.
Which is why purebred dogs are so crazy, right?
Yeah. You know, because especially, yeah, when they were, if there's a way of cloning, maybe that's better. But if you are inbreeding them too much, I think at a certain point, it will lose its, all those genes that you're trying to maintain, it will run out. I mean, you can maybe do 1 or 2 crosses, but if you keep doing that, you start having bad things happening because of limiting the gene pool. All right? Yeah. So that's how that is. So variation is very important. All right? It's extremely important for that reason. So this is one of the things that results in having more variation is the crossing over, So now you have this and the independent assortment. All those things lead to different numbers of or different genetic material in each gamete. All right? So crossing over in them is very important, and it limits the occurrence of recessive traits. All right? All right, so this is the basic stuff about dominance. So a dominant gene will be shown in the phenotype, over a recessive gene. And it's normally uppercase letters. Say for albinism, that's or skin color or whatever, it's going to be S, big S, and then recessive trait will be small s. Okay, so anybody carrying a dominant gene for that on a pair of chromosomes, that will be shown in preference over, and it's masked over the the recessive traits. Then with that also comes a condition called codominance, where let's say you have 2 dominant genes for skin color, for instance. Let's say you have S1 is dark skin color, and then another dominant gene for skin color, which will be S2 light. So if somebody inherits the S1 and the S2 gene together, they're both dominant, right? they're going to be blended. So that's what codominance is. So that you have somebody who's somewhere in between the two. All right? There's another condition also called sickle cell anemia. Sickle cell anemia, which is malformation for the gene of what compound in red blood cell? Hemoglobin. Yeah. It's a deformed hemoglobin that results in a sickle-shaped red blood cell, like that. Right? And then, of course, they become anemic. They can't distribute what compound in the body properly? Oxygen. You know, they become tired. Muscles don't have oxygen. All right? So it also has it's a codominant trait. All right? So if somebody inherits a gene for sickle cell, okay, sickle cell. I can't keep on using S. You know, but I will use it. Okay, sickle cell A, and then a normal gene. Okay, so they're both dominant, right? So they're going to be codominant, and they'll get a sort of half-baked condition, if you will, or somewhere in between, called sickle cell trait. Okay, it's not quite sickle cell, but it's sickle cell trait. You know, and there's a little bit of that. So it's in between two. Okay, so it's diluted. So codominance does that. It will dilute the effects of two dominant genes that are inherited at the same time. All right? All right, there you go. And then, of course, recessive is the opposite, and recessive genes, they are masked over. The only time when you can show a recessive trait is when you inherit what? On both chromosomes, you inherit the, like for albinism AA, you inherit that.
Or if it's X-linked and you're a male, you exhibit it too.
Yeah, if it's X-linked and you're male, because there's nothing on the Y to oppose it. Okay? Because the Y chromosome in the male, we'll talk about it later on when we talk about sex-linked traits, pretty much only has male characteristic genes on it. Whereas the X chromosome, because it's longer, it's got the female characteristic genes on it, but also it's got other traits on it, non-sex traits on it, all right, that the females benefit from. So recessive conditions only show, and these are recessive conditions that are prevented from showing by genetic variation and crossing over and all that when you have a wide gene pool, okay? All right. Okay, so this is inheritance of albinism. It's really better signified, we haven't talked about it, but we'll talk about it in a little bit, by a Punnett square. It's a recessive trait. Punnett, is it double T or one T? Whatever. But it's a Reginald Punnett is a, English geneticist. So what you do, so that you don't crisscross and you have all this confusion on it, for the mother, so if you have the mom on this side, you've got an A, because A is a carrier, right? They exhibit normal phenotype, but they carry the recessive gene, okay? Because this is dominant, so they will show normal skin color. They will not be albino. And then A here, okay? And then the dead on this side, all right? This is an A, a small a, all right? So the dead will have, I'm just going to put it on the bottom, okay? We'll have what? Another A. And then we do that. Oh, my A keeps looking like a 9. Because I'm going to have a question on Punnett square in a little bit. All right? Okay, so what you do, you just do A, right? So this is AA. And then this one will be A, so that's a carrier. All right? And this one will be AA, so that will be, what condition would this be? This person will be an albino, right? This will be this guy, all right? And then A and A, So you have a quarter albino, that's the frequency, or 25% will be albino, and then 50% will be carriers, normal carriers, right? Because in a little bit I'll ask you about, frequency of, these inheritance of these traits, so it's good to know how to use the Punnett square, all right? But anyway, I just wanted to show you how you could transform all that into a Punnett square, which makes it so much easier. And then, of course, 25% is dominant, normal skin, no carrier. All right? Okay, good. Question? Okay. Start polling. Oh, stopped. Show results. Yeah, we got everything on this one. Yeah, but let's see. Why did it jump? It went all the way. Maybe next time I'll just go and hit it on the body to work. So, yeah, it's red-green color blindness. That's a sex-linked trait. So it occurs on the X chromosome, all right? And then we will do a little bit of crosses so we can see, using the Punnett square, how you can inherit sex-linked traits. All right. So Punnett square shows the probability of inheriting a genetic trait. All right. Because later on we'll be talking about pedigrees. Right. Oh no. I could hit. Oh no, crap. Too many buttons. Sex-linked traits. Again, going back to our favorite females, they get the X chromosome, which is longer, and we said the X chromosome carries not only the sex genes, female sex genes, but it also carries non-sex genes. Okay, and those can be inherited as sex-linked traits appearing on that. So for the female, we'll have XX, and then the male will have XY. So if there's a recessive trait on here, like what you mentioned earlier on, George, then on the male, there will be nothing to oppose it on the Y. Okay? And then so males will be more likely to show up with these recessive traits, the sex-linked ones, like green. In fact, my advisor in college was red-green color blind. And it so turns out that the red light and the green light is, the lights. So I'm not sure why they chose those colors, because, if you're red-green color blind, you can't tell what color it is. It's the light, So I don't know why. But I guess red and green you can see, right? Huh?
So that'd be hard during driver's ed.
Yeah. But I don't know how he used to drive, though, come to think of it. My advisor, he was red-green color blind. So when he's driving, but he used to drive okay. Oh, they know that red is on top, blue, blue. I must be red, green, color blind, eh? Red, amber, and then, no. Red, green, amber? Is that right?
Red, amber, green. Supposedly, and I only say this because I keep failing the dot tests.
Yeah.
I've got that red-green color blindness.
Really? Yeah.
However,
yeah.
Now, I don't know, it's not like something where all I see is gray in place of those. It's actually, the way it was explained to me was that the way that I see red is not necessarily the same way
that others see red.
Or
okay.
Someone else.
I was going to say, come live in my neighborhood. We only got one light. And then you don't have problems with that, you know what I'm saying?
I see what I represent as red.
Yeah.
And what I see as green.
But as long as you can see it, because it just was recently put on there too. We didn't have any at all, no lights. Yeah, it's a small town, 3,000 people. So that's the so yeah. Yeah, I know. I've always wondered how that happens. Yeah, so that's what happens. And so anything that's on the X chromosome or number 23, will be a sex-linked trait. Sometimes they call it X-linked because it's on the X chromosome. So so X-linked and sex-linked, same thing, right? The male has got, of course, the Y chromosome, and that is much smaller than the X, all right? And it only contains genes for male sexual characteristics. Okay, they don't have any of those other genes that the females have at their disposal, So anyway, so that kind of marks the differences. Between the two guys, what happens is that sex determination, since the female has got XX and the male has got XY, so the sex of the offspring is determined by the male, because the female can only give X, So it depends on what the male gamete comes up. Okay, either it could be the X1 or the Y1, right? And it's 50-50. So you have an equal number of male and female, right? I had theories about how, if you want to have a boy, they said the Y sperm swims much faster. So you've got to have sex just before ovulation, all right? It swims much faster because it's using all that energy. It survives in the female reproductive tract shorter, okay? But if so if you have sex very close to ovulation, most likely you have a boy, all right? If you want to have a girl, then you kind of have a few days before, because the X gamete from the male swims much slower, so it conserves energy, and so it gets there later, though. Okay, but I think it's just because of the 50-50 thing, unless if you're in China, But you could try it though. If you want to get for a boy, you have sex just close mid-cycle. If you know your cycle, the females will know. You can try and see. Maybe you get a boy. Who knows? But I think it's mostly 50-50. But in communities like China, with the most populous nation in the world, they have a fifth of the world population, right? So they limit birth, and so what happens is that people want to have boys so that they carry on the name of whatever, So you end up having a skewed ratio when humans mess with it. But otherwise, in other times, you'd have 50-50. 50% girls. In China, they actually, fight to find a girl because it's all boys now. It's all been messed up, that ratio, because of the selective, birth. If they have a female, they kill them. I mean, really. Sometimes, or they abort them, or whatever the case may be. Because they only have one kid, so they want to have a boy, and then those boys don't have anybody to marry. They didn't think about that, I guess. That's a good way to kill your race. I mean, maybe it's because they're the most populous world. They kind of need to reduce a little bit. So, so if they're all boys, otherwise they would have to breed with other races to, you know what I'm saying? And that you start to kill your own race that way. They didn't think about that. Yeah. Oh crap! I gotta erase this stuff. And there you go. So and the karyotype shows it. The XY, which is much shorter, and of course the female. And then the pair will make the 23 chromosomes will make the 46 at unity. All right, when you form the zygote. Oops, I got it right here. So, like I was saying, all the non-sexual traits are carried on the X chromosome. All right. And those non-sexual, non-female sex characteristic traits on the X chromosome, those are what are called X-linked traits. So if you have a recessive, gene on that, then you get that trait. If you're a female, we'd have to have both X's with a recessive gene on it to show these non-sexual traits, which are called sex-linked or sometimes X-linked. All right. Again, this is just showing you sex determination, and we just talked about it recently. The male is the one that determines the sex of it. You know, the swimming part of it, I don't know about that, but it's mostly 50-50. There's a 50% chance it's going to be a girl or a boy. All right. So if the Y shows up, that's what determines the maleness. You're going to have a male. And we'll talk a little bit about some conditions where if a male has a Y and then 2 X's, you then have problems. And we'll talk about that. And we'll show the karyotype and the phenotype as well. Question. Now, this one is really involving. Maybe I'll give you a little bit of time to look at it, then I start the polling. Because if I start the polling, you may not have enough time. So this is a sex-linked trait. It's on the X chromosome. So I'm going to start polling. So you draw for yourself the gametes for the mother, the gametes for the female, and you could do a quick Punnett square like I showed you that time. And then, you know, and just make sure that it's on the X, you know, and, you know. So I'll start the polling. And the mother is not a carrier though, right? So it's got dominant, both dominant. Ooh, just press. Okay, so we got a mom here. She's not a carrier, she's colorblind. So we're gonna say mom X, oh crap, why is it doing that? X and then X. So we're gonna put a little C there for colorblind. Oh no. It's normal. So we'll leave it like that, okay? We'll leave it well alone. And then the dad who is colorblind. So it must mean the dad is got a small c, colorblind. If we can actually put big C's on this. Since she's not a carrier, so we know exactly what's going on. And then, of course, the Y doesn't do anything. So that's why the father is colorblind, because you got the recessive on the Y. All right? So for girls, they must have which kind of gamete from the dad? X. So, and they only have X with a big C from the mom, because it's going to be a girl, right? So 100% of the daughters will be carriers. You see that? So I'll show the answer, right? Oh, no, crap. I got to show results first. Oh, no. Oh, Lordy. So, what's the answer here? 100% of the daughters will will be carriers for color blindness, right? Because they can only get a big X with a big C, and they can only get the X with a small c from the. So you will have 100% of the daughters will be X, big C from the mom, X. So they'll have normal phenotype, but they'll be carriers for color blindness. You see that? So how many got this one right? Yeah, this one is a bit challenging. So yeah, you know, 100% of the daughters will be colorblind carriers. All right. So answer. Okay, so that's the answer right there. 100% C. Did you see how we got that right? It was a bit challenging, but yeah, you know. Once you learn to use the Punnett square, it's really easy to determine this probability of the genetic, inheriting these genetic traits, whether they be sex-linked or normal, you know, non-sex-linked traits. All right? So 38% this time. Okay, no problem. Hide results. And next. Okay, so there's another one. So I'll do another Punnett square for this just so you can see. So this mom is a carrier in this case. You see, it's got X and then a little thing on the X so she carries the recessive trait. So she will have one of the gametes will be X with showing the recessive trait, and the other one will be X with normal. Okay, and then the dead, of course, is normal. So if we do the Punnett square, we get the dead on this one. So the dead is X, Y, right? So we do that. And then the mom on this side. So whenever you do a Punnett square, you just do this quick X's and O's kind of thing. So the mom is got a normal X, all right? And then and then what else she got? She's a carrier, all right? Oh, it's like that, eh? Okay, so we do that. Okay, so this one will be XX. So that will be a normal girl, right? Okay. And this one will be, so this will be a carrier girl. Carrier, you know. English is very hard because they got the same words, two words, it is almost pronounced the same, different spelling, different meaning, so it's hard. And then, so you got X here. So this will be what kind of phenotype? Normal boy, all right. And then this one, because I think I have another question on the sex-linked traits. One of these questions. So if you get used to using a Punnett square, that'll be good. And then you got X here, and so this boy is gonna be having that condition, whatever the condition is. All right. Okay, so that's how you. This one is kind of confusing. It's like things crossing, and the Punnett square is the only way to go about it, so you can tell what's going on. All right, so I can use this too. So genetic mutation, there's a change in the genetic material. The base pairs are no longer aligned the same way, and when we talked about it earlier, I said, the codon has got 3 bases in it, and if you remove one, everything goes out of position, and it will code for a different amino acid. An amino acid will have maybe several codons, that triplet code. So if it's changed, it might code for a totally different amino acid with a totally different R group, right? So, and the way the protein folds up, whether it's going to be a globular protein or a fibrous protein, depends on the R groups, because R groups will attract each other when it folds up. All right? So genetic mutations, of course, can result in abnormalities and cause disease. And later on, when we start talking about genetic conditions, we'll talk about disease genes. Like I said at the beginning, there's really not a disease gene. It's only when it is a mutation that's when it can cause problems. Otherwise, genes normally code for, for good traits. It's not like there's a gene that is specifically in our body that will cause disease at all. All right. And earlier on, I wanted to talk about genetic engineers and agricultural scientists and how they manipulate the genetic code so that you can improve the products, agricultural products, and so forth. Right now the hot topic is the GMOs, which are genetically modified organisms. All right, this is supposed to be bad for you, right? But when you think about it, what are these geneticists and scientists trying to do? Yeah, what they're trying to do is switch on certain genes that are actually good, that have good traits. Okay? They are trying for the organisms to show the good genes. So do you think that is good for you or bad for you? You see? So the controversy about this, I think whenever humans don't know anything or a lot about something, it becomes controversial automatically. Yeah, that's what happens. So I think with this GMO, until they know a lot more about it, then you could actually say, are these genetically modified organisms really bad for you? Because now if it doesn't have GMOs, they say it's good, it's a good product. But I think it's just because they don't have much knowledge about, you know, how you switch on genes. Because what they're really trying to do is to switch on the best genes so that they are shown in the phenotype. So when I'm thinking, just like a regular scientist, I'm thinking that's good for us. You're getting organisms. It's like adaptation. When we adapt to certain conditions, we show up a gene that actually, you know, makes you survive in that condition. So if the geneticists are switching on the best genes in that organism and getting the phenotype out, that could be good for the organisms, right? It should. Don't you think so? I mean, I could be wrong, but just reasoning like that. But they haven't kind of proven it yet, so there's a lot we don't know. I'm just trying to get you to think about it. You know, that's what I'm trying to do here. I'm taking a stance so that you may say, no, hell no. You know what I'm saying? The thing is, when you think about something, you know, when you're inquisitive about something, you learn about it. That's the only way to you know, to increase your knowledge. If you're not inquisitive and you don't want to, you won't learn, even if you're brilliant up here. You know what I'm saying? So anyway, so that's the one thing I forgot to mention, and I really wanted to talk about that. I got it. Yes? We got to do what now? Oh, you got a bathroom break? I've put in a break somewhere. It's the middle slide, so I guess I'm going slowly. Maybe I should speed it up a little bit. Less stories. Do you want to? Because I think I have how many slides? I've only got 2 more slides, and then we take a break because I've got to. And then after that, I'll really go. Well, not too fast. Okay. So sorry, guys. I didn't think about that. Thanks. You know. I thought that the break I put it in the... Oh, we got a question. I hope you can get this question right because you kind of feel like peeing right now. Okay. Science. Okay, so I'm going to start the polling. And then we'll be talking a little bit about that. So we can make it fast, and then you can go on a break. Yeah, so after this, I just have the one. Okay, very good. Show hide results. Yeah, no, epigenetics, yeah. So 59% got it, which is good. Yep, that's the answer. This is a condition where, you know, well, it's a science about how the environment and behavior interacts with our genes to show certain phenotypes. Things like cancer and something like that, they also fall under the epigenetics because sometimes you may have a predisposition for cancer, even when you talk about twins. And then one twin, because they're exposed to a certain environment, may show the cancer, and the other one who's not exposed to that environment will not, even though they have the genetic predisposition for it. All right? So I only have this one slide to go, and then boom, we go for a break, you know. After that, and then after the break, we'll be talking about these single gene diseases. It's a single gene mutation for a particular one protein, okay, whether it be hexosaminase or whatever the protein is. It occurs in one gene or a group of genes. All right, and then it can be inherited. It can be passed on to other generations. All right, and some diseases result from this combination of inherited genes and environmental factors, like I was talking about. Sometimes, if you are not exposed to the kind of environment, you won't show that problem. But if, like radiation, if the one twin is irradiated, twins. I'm using twins because they've got the exact same genetic material, right? If they're identical twins, that is. All right. So one is exposed to certain condition, and one is not. The one who is, or the one who smokes, for instance, chemicals, is the one who's going to get lung cancer, and the one who's won't get it, right? So that's what it does. What is epigenetics? It imprints into the genetic blueprint a certain trait. Like if parents were to eat a certain chemical or whatever, or in their diet, and it affects what is called methylation, which is addition of methyl groups on the DNA. Methyl groups are like this, and those can switch on and off certain genes. And when we start talking about cancer, that will be very important, because if you are over-methylated on your DNA, you can switch off certain genes that actually inhibit cancer, and then you get the cancer. All right? And it could be an interaction with the environment, and it depends on what diet you're eating or which conditions you're exposed to. Like if you live near the Hamptons, what's it called? The Hanford site, you know what I'm saying? And then you'll have more because of the nuclear radiation. All right? And then, great. Sorry guys, I really apologize. So but we'll go a little bit faster next time, okay? Okay, guys, thank you so much. We're going to proceed. We'll talk mostly about the genetic diseases for the remainder of this lecture. And I hate to do this to you guys, but question. But this is not so bad, So I'll start the polling. The last two guys. They abstain. So trisomy, of course, it's a chromosomal condition when you have one of the chromosomes pair doesn't separate during meiosis, and then so you'll have both pairs going to one gamete and the other gamete getting nothing, and the other gamete that gets nothing will get monosomy, which is what we're going to talk about, because they'll get the other gamete the other part of the chromosome from the other parent. All right. So trisomy, of course, you will have an extra. Instead of having a pair, you have 3. So how many? Yeah. So a bunch of them got it right. 70. Yes.
Do you have a trisomy of like multiple chromosomes, like 321s and 322s?
You don't, but you have that condition, and it's called a chromosomal condition. The one I talked about is tetrasomy, actually, of 18, where you have 2 pairs of it. Like you have 2 pairs of 18. One is a normal 18 with the short and long arm, and the other one is one with 2 P's. So you have 2 of those chromosomes. So you have tetrasomy, so you have 4. So in that individual would have 48 with tetrasomy. So yeah,
He's asking if you can have 3 for 21, and then also 3 for another, for 18.
Oh, okay. Do you know what happens? Do you know what happens in nature? What happens in nature is that when there is heavy genetic malformations, those are aborted naturally, and the female won't know it. Once that gamete is formed and it's got all sorts of double up chromosomes, it never survives, and they will just have a normal period, just like they do. They may have been they won't know it. So that's the good thing about nature. If the deformity is too much, it won't survive. That zygote will never be. So you will never get that kind of deal where you have double up on this chromosome and double up on another chromosome. See what I'm saying? Nature is a way of correcting for that. Oh yeah, good. It's a good thing, because I had not kind of gotten it. So anyway, so this is 47, right? So, oh, there you go. That'll do. So most of you got it right, so that's good. Trisomy is because one of the pairs didn't separate during meiosis, and then you get 47. All right, good. So we hide and proceed. Yeah, so these are the chromosomal diseases now we're going to talk about. So the result from when either the chromosome could literally break, and so that you don't have all of the chromosome there, or the chromosomes may fail to separate. Okay, the chromosome pair So you have trisomy, like that one. And that failure to separate during meiosis that's what's called non-disjunction. All right? Failure for those chromosomes to separate is non-disjunction. All right? So you'll end up with a gamete for that particular chromosome. It will have the 2, and then another one will have nothing in it, because the pair of chromosomes went to one gamete instead of going one to each. All right? So this gamete will produce what's called monosomy, because it will get the other part of the pair from the other parent, and then it will just have the one. And this guy will get the other pair, the other matching pair from the other parent, and will have 3. So this will be trisomy, and this will be monosomy, and they result from non-disjunction. Okay. Based on our knowledge of meiosis and cellular organelles, what do you think could cause non-disjunction? Yeah, centrioles, very good. Centrioles, remember the centrioles are the ones, they are rod-shaped proteins that will move and sort of scaffolding and so forth, move the chromosomes to the center. So if, for instance, let's say you're much older. When you're much older, everything kind of slows down. And your protein-making machinery is also slowed down. So you don't make enough centrioles. That's why when you're older, the chances of you getting Down syndrome is higher. If you are older than 40 females, the chances of getting a Down syndrome are much higher. That's trisomy 21. It's trisomy of the chromosome 21. Okay? So if you want to have your babies before you turn 40. Not to say that when you hit 40, you always have a Down syndrome. I mean, it just means the likelihood it gets much higher. And that could be attributed to those things that happen when the cell is not functioning properly and all the organelles are kind of doing their thing. So non-disjunction can happen because of that. And also spindle formation. When you're forming those spindles, if you don't make enough of the proteins that make up those spindles, some of those chromosomes are not going to be dragged to the center so that they separate. They're going to remain to one side of the cell, and then when the cell divides, you get this condition. All right? So I want you to start thinking about some of the things, what parts of the cell could go wrong to create some of these conditions. All right? Okay, so next. And this is like pretty much a karyotype showing where all those conditions occur. And you can see on number 23 here, all these things like muscular dystrophy, it's a sex-linked condition. Of course, you know, all these neurofibromatosis, you know, no, that's not the one. And hemophilia types A and B, and also male pattern baldness. It's also sex-linked. Recessive trait, like me. So that means my father is bald, but I didn't get the condition from my dad because it's sex-linked. I got it from my mom because I only got Y from my dad, and Y doesn't have anything on it. So it happens that my mom's family is also bald. So she had the gene for it, so that's why. My friend there, Jim. He didn't get it from your dad, you know, because if I stand my dad over here, we look alike and we both got bald. They say, oh, you must have gotten it from the dad. I didn't get it from my dad. Got it from my mom. See how funny it is about those things. So this is just a karyotype showing all those conditions, so we just go quickly on that. So effects of non-disjunction, like I drew earlier on, you have the mom here. So one of the chromosomes, it could be any of the chromosomes, you know. If it's 21, then you get the Down syndrome. So one ovum will get 2, and then the other one will get nothing. This one that gets nothing creates monosomy because the other parent will contribute the other gamete for that particular chromosome. Okay. Usually it occurs on one chromosome. It doesn't occur on multiple chromosomes because I think those zygotes are aborted naturally. And then, of course, the trisomy. All right? So that's non-disjunction there causes. So you can all explain non-disjunction and how it causes trisomy.
Men and women, non-disjunction?
Yeah, that's a good question. I didn't think about that. But I think it does, but I think it may happen more with the X chromosome and maybe more with the females. Okay, because we have a condition where the offspring gets 2 X's from the mom. That means the mom must have had non-disjunction and gets a Y from the dad, you know. So I'm thinking it's more prevalent when I just look at the diseases that we're going to talk about, and those diseases are kind of representative of the whole gamut of these genetic diseases. For the most part, you get the 1Y, and you get multiple X's. For the most part, for all the boys, like the Klinefelter syndrome that we're going to talk about, you have 2 X's and 1 Y. So I would hazard a guess that it happens more with the females. What's that?
The X could come from the dad too, right?
Yeah, the X can come from the dad.
If you have 2 X's and a Y, it's not always come from the mom. If you have 2 X's, it could have
No, there's only 1 X from the dad.
Because the gamete is haploid.
But if they didn't split,
doesn't split.
if the dad's XY didn't split,
Then you'd have XXX. Superwoman.
Yeah, that's a good one. I haven't seen the condition though where the dad doesn't split, so you get XY from the dad, and then you get the Y. I think it happens on the X more because the Y is shorter. Remember, the Y is a shorter chromosome, so it's most likely to happen with the X part, and it's going to be the non-disjunction. I'm going to look it up though. I think I would hazard, because just by the way it is, and the X being shorter, because it doesn't have anything to oppose it. The Y is shorter and the X is much longer. I would think, but that's a good observation right there about whether the non-disjunction happens equally in males and females. So that's homework for next time. You guys research it out and tell me about it. Yeah. But I'm thinking it's more common with the X with the Y. Because I don't think
Based on size alone?
Yeah, because I'm thinking and sort of mechanically how that could happen. Okay? I'm thinking the Y, because it's a shorter one, it's most likely gonna, but the X, because it's not. When you look at the karyotype for the male, you will see something like that and something like that. Okay, if you see the karyotype for the female, I think you will see this with the P and Y arms. So that those are the ones that can have non-disjunction, whereas those other ones are separate. I think that's the case. We'll look at some karyotypes very soon. But I think that's what because it's shorter, it's not really paired, it's not stuck to it with the centromere. You see what I'm saying? Because there's no way to stick it on. You see? For you to have a short and a long arm. Yes?
Can non-disjunction happen also when the crossing over happens and they pair up and then they don't unstick?
When crossing, so crossing, yeah. And I'm thinking, because crossing over.
What happens in trisomy 21 when they come together? And then
And then instead of exchanging genetic material, they really like each other and they stay together. Yeah, that could be. But then another way of that happening is when the spindle formation, the centrioles and all those things are not working properly to segregate the chromosomes in the center so that they can both separate. So that's definitely a way, because at that time they're really close to each other, they're really close proximity to each other. So to actually answer your question, the X and the Y are not attached by the centromere because of the size. So non-disjunction cannot occur because they're not attached at all in the first place. You see what I'm saying? You will see the karyotype very soon. Because there's no way to kind of put a centromere on them like that. But with the X, with the female though, because the X, they're both same, and then the centromere will be right here. So you have your P and your Q arm on the X. You will see on the karyotype of the... so that's a good one. Thank you. And we got the answer right away too. So that's good. All right, very good. Oh, question. I put too many of these things. So, which genetic disease results from the recessive gene that fails to produce that converts to tyrosine? Yeah. So it's a single gene disease. Oh, start polling. Also. on, guys. Punch any answer. Okay, stop polling and then show results. Phenylalanine is converted to tyrosine by phenylalanine hydroxylase. So it is phenylketonuria, right? So that will be A. So a bunch of you got it. Great. Oh, it starts where I left off. Oh, I see. So yeah, it's and we'll be talking about this condition in a little bit. So that I'll just go right there, so we don't waste too much time. So it's A is the answer. Okay. So these are single gene diseases where you have a problem with the sequence of one gene. And one gene, we said the final product is a protein. So you make a defective protein that doesn't do its function. All right? And the first one is cystic fibrosis. That's the first condition we're going to talk about. And this one, it's transmembrane protein. They call transmembrane conductance regulator. And it's sodium and chloride ion channels. These are transmembrane proteins that transport sodium and chloride in and out of the cell. So what happens is that in cystic fibrosis, that protein, is defective and it's not transporting the sodium and chloride. And what happens is that exocrine glands will produce very thick fluids, like sweat glands are exocrine glands, and mucus, cells that produce mucus in the respiratory system, in the digestive system, will produce very thick mucus, especially in the respiratory system where it starts to clog up the respiratory system. All right, so that's cystic fibrosis. It's that transmembrane protein, transmembrane across the membrane ion channel. So the gene for that protein, which is occurring across the membrane, cell membrane, will result in that position, in this condition, sorry, and then you produce, and then later on we'll be talking about gene replacement therapy and gene augmentation and so forth to treat some of these conditions. But first we'll go through all the conditions, and so I'll need you to know the protein that is a problem for these single gene diseases, and what the function of that protein is and how that condition comes about. All right. And then the treatment, which we'll talk at the end. Sometimes they use a cold virus, because the cold virus, attacks your respiratory system, and then it will inject the good gene for that transmembrane protein, so that your cells will express that, and then you won't have. It's like gene replacement therapy, which we'll talk about in a little bit, and then you start having the normal protein, and then you have less thick mucus in your respiratory system, and then less obstruction, and and so forth. You could die from this actually. So, yeah. And this is the one that we had a question about, phenylketonuria, where you have an accumulation of the amino acid phenylalanine. It's got a benzene R group on it, like that. It's got. Okay. And then that's the R group on it, and then of course you have the N H3 here, and the COOH, and then the H over here. So that's the amino acid which is converted by phenylalanine hydroxylase, which is the enzyme in question that the gene is a problem, and then phenylalanine hydroxylase is not catalyzing the exchange of phenylalanine to tyrosine, which was the question we just shown. All right, and then it accumulates in the brain. Phenylalanine, it damages brain tissue. You get mental retardation, and you could die. So the accumulation of it could cause problems that may result in death. But things like aspartame or sweeteners that have phenylalanine in them, you have to avoid them. That's why they always put that note on there, so that you don't take sugar-free, like my thingy here. Is 0. If I was phenylketonuric, I wouldn't be able to use it. It's not, what do you call it? Bud Light. It's Sprite. Most people confuse it for Bud Light. You know, so I have to put a disclaimer. Oh, it's Sprite Zero. Okay, so when you have an accumulation of phenylalanine in the system, what happens is that it gets converted to a compound called phenylketone, which then goes into your urine, hence phenylketonuria in urine. You know, and at birth they normally try to check if that gene is on board, that recessive gene, so that you can have a diet plan for it, in terms of how to treat it. All right, so you avoid anything that has phenylalanine in it. All right. Okay. Question, because we're going to be talking about these conditions in a little bit. So which genetic disease is caused by a recessive gene that fails to make essential lipid processing enzyme? All right. Start, Paul. All right. Okay. Show results. Yep. It is Tay6. It's a hexosaminidase. It's tongue twister for me. But that enzyme it processes some certain lipids, so you get an accumulation of these lipids again in the brain. It causes severe mental retardation, and by age 4, usually death. All right. So that's Tay6. Yep, Tay6. Okay, very good. So we will find results next. All right, so more examples of single gene diseases. So Tay6 is the one we just, Tay6 disease, TSD. It's a recessive condition, all right? It's failure of this hexosaminidase. Yeah. The names of these things. Hexosaminidase is the enzyme. So, you know, that is, it's got a problem gene, you know, and it's not processing these lipids, and then when they accumulate in your brain, you get problems. All right? All right. Yeah, like I say, you know, it accumulates in your brain, all these lipids, and then, you know, can result in death by age 4, Tay6. All right. There are certain Jewish communities that have a high prevalence of Tay6. Does anybody know what reason that is? Certain Jewish communities have a high incidence of Tay6 disease. What is the reason that geneticists or epidemiologists attribute to that? Was it? Yeah, that's right. What happened? Who has resistance to TB? How? Why? Which kind of Jews? I mean, I don't mean like, but what kind of phenotype, genotype?
The carriers?
Oh yeah, the carriers for Tay6, it seems, they have a very high resistance to TB. Right? And, you know, back in the day when they used to put them in concentration camps, the poor Jewish communities, they were so crowded. And you know how TB is when you're crowded. It's an aerosol thing, and they get it. So the carriers for Tay6 have a resistance, so they survived. Right? And so then they would continue to, you know, populate the place, and then you end up with a whole bunch of the Tay6 because they're carriers for it. You see what I'm saying? So you would have like a T and a small t. So they're carriers. So they will end up with at least a quarter of their siblings will be having the Tay6. So that's how it became prevalent, just because of the way they were concentrated. And, you know, the TB, turns out the Tay6 heterozygous condition, you know, is more resistant to TB. Okay, so anyway. There you go. Very good. So epigenetic conditions, you know, result from the interaction of, you know, behavior and the environment with the genetic makeup to result in certain conditions. And like I was talking earlier on about cancers, certain cancers, what happens is that, if you have a high level of methylation, like I was talking about, these C groups on your DNA, or sometimes low levels of methylation, it can switch on and off certain genes. So it happens that if you have a high level of methylation on your DNA, it will cause switching off of genes that prevent cancer development. Okay? Because one of the things you guys need to understand is how cancer comes about, as a result of the interaction with the environment, the genetic makeup, and, some of these chemicals, all right, and what happens to the DNA. So methylation, under-methylation or over-methylation, and normally you get this kind of codon being repeated. CGG, C-guanine, guanine. If the longer that repetition is on that gene, okay, because that's the sequence, that's the codon that if you have a string of them, they switch on certain genes based on the length of it. You see what I'm saying? So if it's too long, it switches on and off certain genes. And it happens that if it's too long, it switches off the genes of the proteins that normally inhibit the occurrence of cancer, which is uncontrollable proliferation of certain cells. They could be blood cells, they could be anything, any kind of cells. So you get the liquid tumors like the leukemias, or the solid tumors, where you have a lump like breast cancer. Okay? All right, so methylation, I want you to be able to explain how, cancer comes about as a result of this interaction of these factors, the environment and, the genetic makeup. I'm used to that thing there. And then there's another condition called Fragile X syndrome, which is also sex-linked. It's X-linked, and of course it occurs more in males. It's a recessive trait, and causes mental retardation and so forth. It occurs less in females, but in females it occurs mildly also because they've got 2 X chromosomes. So even if they have the recessive trait, it won't show, anyway. So that's one of those other diseases. Okay, let's move on. We got another question here. So this is to do with another disease we'll be talking about in a little bit. All right? So this condition occurs in females with a single X chromosome and resulting in XO. All right? So all right. Okay. Show answers. Of course, it's Turner syndrome. Yeah. So a bunch of you got it right, and. That's good. It's Turner syndrome, where you have. So it's kind of what condition is this? What does it result from? Monosomy, right? Monosomy. Yeah. So this is monosomy. Okay, because you get one with a single, and then another one with nothing, because non-disjunction, both pairs went to one gamete and the other one got stiffed. Okay, so. All right, so next. So chromosomal. So we just covered single gene diseases where a single gene is affected. It's a single protein, like phenylalanine hydroxylase or hexosaminidase. Now we're talking about the whole chromosome. Either it's broken or you have trisomy or monosomy, right? So, and then we kind of said at the beginning, earlier on, Down syndrome. It's a popular condition. Well, it's not popular, but it's it's one of the common conditions that occurs, and it occurs on chromosome 21, and it's sometimes called trisomy 21, when non-disjunction happens. So that's the other name for Down syndrome, is trisomy 21. All right. So you have, instead of having a pair of chromosomes, you have 3. All right. And of course, it leads to a whole host of symptoms, characteristics like, you know, a little bit of mental retardation, physical defects, and things like that. Next. And then this one is also trisomy, and it's Klinefelter syndrome, where the X gametes fail to separate, and so you get XXY. All right, and those males are normally chubby. They've got large breasts and very long limbs, long legs. All right, if Down syndrome is trisomy 21, what is this called? This Klinefelter syndrome. Yeah, trisomy 23, right? Because it's trisomy of the sex chromosomes. All right. So, and then it has also some other pulmonary problems with it as well, you know. And then later on, I'll be showing you the phenotype and the karyotype, and then I want you to be telling me which condition it is before I bring it up. All right. This one, which condition is this? Yeah, trisomy 21. Do you see? Do you see our X and our Y here on this one? They're separate. Okay, and I was going to have somebody come up and circle the problem part. So maybe we'll have somebody come and circle up the problem part in the next. How about that? All right. So yeah, this is Down syndrome. Okay, so this one. Okay, does anybody want to come and circle the problem on the karyotype? You can use your finger.
For the sake of time.
Yeah, for the sake of time. I mean, you don't have to. I mean, yeah, yeah, yeah. So you got the double X, you see, and then instead of having one X, and then of course you've got the Y here. Okay, so that's it right there. Yeah. Did you have something there? You know, no, you're stretching. Okay. We're almost done here. So that's Klinefelter syndrome. Turner syndrome. Oh, crap. Oh, yeah, we'll talk about it. I don't have a picture for it. Okay, so, but Turner syndrome was in the question. It's monosomy. You have, you know, XO instead, and it happens in females. All right? And they have kind of like a webbed neck, a very short stature, of course, sterility, you know, those females, and sort of immaturity. They don't mature sexually, you know, and so forth. And this is happens with, you know, when you get non-disjunction on the 23rd, of course. All right, so that's that one. And I think I'll show you a picture of it, too. I will. And then, yeah, there it is. Okay, and the problem, so that we can go fast, of course, is Turner syndrome. You see, 1X, and of course there's no Y, you know, so it's 1X, so that's where the Turner syndrome. So if you see a bunch of these karyotypes, being the doctor that you will be, you can say, okay, this person is this person is that condition just by looking at that. See what I'm saying? So you have to know which chromosome is affected, and you know, and what condition it is, with trisomy, monosomy, or whatever. All right. Okay. Question. We're almost done. I promise. Okay. Because we'll be talking about this in a little bit. So we have a question. Start polling. Somebody told me that you guys like this, so I may have went overboard with the questions. So sorry. So I was like, oh, if they like it, man, I'm going to just dish it out. So over several generations, we did a Punnett square, and we talked about the Punnett square, and we said it shows the probability of inheriting certain genetic traits, right? But of course, this would be the pedigree, which shows over generations how a trait could be, you know. And I'll show you a pedigree, and we'll try to sort of determine what kind of thing is going on there, okay? All right? So there is that. Oh, I didn't show the correct answer. So the correct answer, of course, is pedigree. Yes, pedigree. Hide. Okay, so these are the things that we use when we're doing genetic counseling. So there are people who are certified genetic counselors, so that they can say, okay, in your family, you're most likely, sometimes couples, because they may have had problems in their family history, they go to a genetic counselor so that they can tell them. And the tools they use, of course, the pedigree, the Punnett square, and so forth, to show the probability of coming up with certain conditions and things like that. And then later on we'll look about at things, how you can determine conditions in utero and so forth. All right, and what ways you use to harvest the cells for that. So anyway, a pedigree, we just talked about it. It illustrates the occurrence of traits, genetic traits over several generations, whereas the Punnett square will show the probability of a particular trait occurring, okay, or of inheriting a particular genetic trait. All right, so. So this is a pedigree here, and the squares are males, all right, and the circles are females, right, and shaded parts are the abnormal phenotype. Okay, and of course the half would be a carrier. All right. So when you look at this, what do you think might be going on in this pedigree? Yeah, it's a recessive sex-linked trait. Do you see how all the females seem to always be carriers, and only the males? Of course, the males can either get a normal one, or they could get the recessive condition. So you see, the condition is only, all the half-shaded females are the normal carriers. They exhibit normal phenotype, but their genotype is that they're heterozygous for that recessive condition. All right. So very good. You guys are good. It's X-linked. Or sex-linked, if you like, you know, whichever floats your boat. Recessive condition. Okay, very good. You guys are getting good at this. Very good. If it doesn't want to go, what did I do? Okay, let's see if this will work now. I guess not. So this is just a Punnett square, and it's showing different, like we've been doing before. So I'm not going to belabor this too much, right? You know, you've got a carrier here, and then you do that deal, and then you get your probability of occurrence of a certain phenotype, right? So we've kind of done this ad nauseum. We'll move on. All right. So a karyotype, of course, is got you know, the picture of that genetic information in the cell of all the chromosomes, and you can determine all these chromosomal conditions. All right. So normally, when a woman gets pregnant, you know, they might want to know if, you know, based on maybe on the deliberations with the genetic counselor, they may be told, you know, in your family you may have a likelihood of creating this condition. So when they get pregnant, they might want to know, okay, what are we carrying, you know, and so they have to harvest cells of the fetus. One of the big ways is amniocentesis, you know, and of course this is done in conjunction with ultrasound, so that when they inject the needle, they're not going to damage the fetus, right? So that's one way of harvesting the cells. For this, I just need you to know the different ways that you know, are used to harvest cells to create a karyotype. All right. This is in utero, that is. Another way that is less invasive is chorionic villus sampling. you know how the fetus is surrounded by the chorion is the outermost membrane, and then the amnion, which is got the fluid inside. Okay, so the chorion, in fact, is the fetal contribution to the placenta, and the mother will contribute the endometrium. So the bag on the outside, for lack of a better word, will be the chorion. So what they do is that they go through the vagina and through the cervix, all right? And because, oh my God. So this is the chorion, and then the cervix, so this is the uterus. And then you have the cervix here, cervix, uterus. Okay, so this is uterus, uterus, uterus. So when they go through the cervix like that, so I could use a different color, the first membrane they're going to get is the chorion, and those are fetal cells. Because the chorion, part of the placenta comes from the fetus. So that's a less invasive way because it doesn't, like go inside and, chance of damaging. And then they get those cells, and then, of course, they take the picture of it, electron micrograph picture, and they show the karyotype, and they can see any kind of conditions that may occur, right? In adults, there are other two more ways of getting the cells. One is, like they use in CSI and so forth. Chick swab, right? So they just take a swab and get some cells, some epithelial cells lining your cheeks, right? And then they could do the karyotype. They could do that. The other one that is used is blood sample with, this is my shortcut for with, white blood cells. Why? Why do you need a blood sample with white blood cells for you to create a karyotype? Yeah.
Yeah, the white blood cells have nucleus. Red blood cells don't actually. True.
Yeah, true. Yeah, you guys are bright, of course. If red blood cells don't have a nucleus, they lose it. So we call them red blood cells in inverted commas. They're not really true cells because they lose the nucleus, and all they have is really a whole bunch of hemoglobin because they need to be transporting oxygen, which is vital. So it can't be bothered with having other organelles. You don't need genetic information anymore. Besides, they are recycled from the stem. I mean, every 4 months or so, you have brand new blood cells. Red blood cells, okay? But, white blood cells are real cells. Those are the only true cellular component in the blood. The platelets are not, they're actually pieces of cells. So you need to have a nucleus in order to take a picture of the nucleus. So that's why. Okay, so you guys got it first. So just know these methods that are used to harvest cells to create a karyotype. All right, so that's good. Oh, my thing is not working anymore. So I've got to do it here. All right. Oh no, I went backwards, I'm sorry. So this is just a picture of, the amniocentesis showing that you would need ultrasound, okay? And the other one would go through here, chorionic villus sampling would go through here. And this, you see this membrane outside? That will be the chorion. And this will be the uterus. All right? And then the chorion will have, of course, its fetal cells. So you can tell whatever condition the fetus has, and so forth. Okay, so how do we treat these genetic diseases? It's a very young science. We still don't know a lot about, treating of genetic diseases. So far, most of the time we've been just addressing the symptoms, but we're not really doing the cure. But right now, several methods they use, like gene replacement, gene therapy, where they manipulate the genes by using genetically altered viruses to inject. They capitalize on the fact that viruses can inject their genetic material into our human cells. So what happens is that you modify these viruses so that they insert the gene that's a problem. All right, and then they use the viruses to infect those particular cells. All right, and they use the cell-to-cell communication and those proteins on the outside of the cell to target that particular cell type. Okay, that needs to show that, and we'll talk about two, kinds. Let me see, where we will we'll talk about gene replacement. all right? And then we'll talk about gene augmentation. And here they just add the cells that are genetically modified, all right, by viruses, and they're added to the to augment, so they produce the actual protein. And then there's some other things like plasmids, which is a circular bacterial DNA that is also modified by recombinant DNA technology and so forth. And my favorite, which we'll talk about also, is HEC, a human engineered chromosome. It's like a 47th chromosome that is inserted, and that chromosome is carrying all these genes that are therapeutic. Isn't that neat? I mean, I really got tickled by that. So, gene replacement therapy, this, you know, we say it with a caveat in terms of replacement. It doesn't really replace the mutated gene. What it does is it will insert the normal gene into the nucleus so that then it will start producing the normal protein. But the bad or the mutated gene will still be there. Okay, so it's gene replacement, but it's not quite. Okay, so but we call it gene replacement. Gene augmentation, on the other hand, is you take, say, what they did with this adenosine deaminase protein problem, which occurs in young kids, and they have this condition called severe combined immune deficiency, where you know, it's like AIDS, really. It occurs in kids, and that protein. So what they did is that they took the white blood cells from those individuals, from those kids, then they genetically modified them, they infected them with the virus carrying the normal gene for ADA, which is adenosine deaminase, and then they multiply them. Multiply until they're 1,000-fold, then they put them back into the individual. That's gene augmentation. See, they're augmenting. They're not changing the cellular makeup or the nucleus of those cells by inserting the gene in there, but they are just inserting cells that have the normal gene for it to produce. You see what I'm saying? The difference, you can see the difference between gene replacement and gene augmentation, right? Yeah, okay, so good. So that's that. Yes?
Can you do like the gene, either one of those therapies in a fetus to avoid like Down syndrome or one of those?
Yeah, that's a good one right there. Yeah, you know, I don't think they can, because you would need entry into the actual fetus as it's developing, and because it's not baked yet, it's not cooked, it's not done. So you can't really modify. You know what I'm saying? You see the problem that can be presented by the fact that it's still in development. You see? So what you might do may be temporary because it's going to change. It's not the final product. So they would never attempt it for that because there'll be just too many variables to talk about. You know, when you do an experiment, what do? You only change one variable and keep everything as constant as you can. Because otherwise, if you have too many variables interfering in it, your experiment is, you're going to go crazy trying to figure out what is causing the change. Right? Yeah. So that's why you would probably never do that. Oh, this is not working. Sorry. This is the one I was talking about, which is the adenosine deaminase, which is that gene for that enzyme that is problematic. And so what happened is that these kids, it happens in kids, and back in 1990, that's when they did the first gene therapy experiment, where they did take out the white blood cells because it's an immune deficiency syndrome, just like AIDS, right? And then they infected them with the virus in this first side of the picture on the left side. And then they cultured those white blood cells, and then they infused them back into those, and it worked, you know, so that they would have the normal adenosine deaminase, okay, because it causes all these problems. And in this other one, they used a plasmid, which is, again, recombinant DNA technology. You can have, it's like a bacterial DNA, and then you modify it and you insert the gene by splicing. You insert the gene for ADA, adenosine deaminase, and then you put it in an aerosol, like a asthma inhale thingy, and then it will infect your respiratory system cells and so forth. You use it like a cold virus to do that, because the cold viruses will target the respiratory system, and then that's another way of introducing that normal gene. So gene technology is excellent, gene therapy. There you go. Oh, my thing stopped working for some reason, but it did carry me on for quite a while, actually. So, and then some more problems here, labor congenital amaurosis, which is a problem with a gene that's found, it's a photoreceptor in the rods and cones, and that gets you blind, you end up blind. So they've tried various techniques where they introduce into the retina, viruses that have been genetically modified so that they can insert that protein. Sometimes they even give you diet per os with the precursors for that protein so that you can make that protein and it may improve vision, because it's a certain protein that's found in the rods and the cones for in the retina for vision. And so there are actually ongoing experiments to get a treatment for that. And then cystic fibrosis, of course, because, like I said, it creates this thick mucus in the respiratory system. So you want to target the respiratory system. What do? You use a cold virus, because what does a cold virus do? It infects the respiratory system. So you can introduce that gene using a cold virus in these patients, and they get well. And they're also doing research on that. On cystic fibrosis. The last and final gene therapy is RNA interference. This is a method of switching on and off problematic genes. Isn't that a neat way of doing it? You interfere with the RNA for that, whatever problem gene is coming out when the messenger RNA or something, you switch it off. And then it doesn't get transcribed into this abnormal protein that's going to cause the problem. So that's another neat way of doing it. And they use it also for research purposes so that they can, in the lab, figure out where the gene is, and, and they can, manipulate it by switching off a particular gene so you can learn what it does and what problems it causes, which helps with the, in finding a therapy for these conditions, right? So RNA interference is another cool one. I like that. And that's it. Thank you, guys. I appreciate it. I did it 20 minutes before time. Thank you, guys. I appreciate it. Yeah. Guys, I will post this lecture on the Evolve. If I can't, I will post it. I'll give it to Linda, and she can post it for you guys on MyHeritage, okay? So you can get the PowerPoint, and also you'll have the ability to listen to the audio as well as see the annotations on the board on integrity, and I'll send you a link to that. That link, I'll put it on the website.
Is there a quiz for this lecture?
Yeah, there is. It's already open right now, and it's due on the 20th.
Okay.
All right. Thank you, guys. I appreciate it. It's nice meeting you. We'll see you next time.